infection diseases, leading the development of genomic technology for outbreak investigations in Africa and South Africa. Dr. Palacios earned his master's degree in biochemistry and his PhD in virology from the University of Buenos Aires. Over to you, Gustavo.
Thank you. Thank you for inviting me to present. And I'm going to be giving a brief introduction about the historic conditions for hantaviruses and spread. So the historical background started in the Korean War with the, essentially the detection of the first hantavirus. And so this was causing hemorrhagic fever with renal syndrome, and essentially the impact was mostly on the kidneys. The second presentation of a different disease, in this case hantavirus pulmonary syndrome, was in 1993 by the detection of Signombre virus, but my friend Stuart Nichols and the team and the special pathogens at the CDC that essentially discover, that hantaviruses in the Americas were producing a different disease. And obviously there is a lot of comparisons between the impact of these two type of presentations and the different symptomatology associated with those. The important aspect is that two groups of viruses from the old world and the new world have a different clinical presentation. So there are a lot of hantaviruses, more than we can count every day. But the ones that we are going to be focusing today is Andes virus, that is one of the members of the New World causing hantavirus pulmonary syndrome in Argentina and Chile. So the distribution of the virus, the diversity of the virus in the Americas essentially have three different big groups. One group in the central region that we call Leishmaniasis virus or Juguetiba virus. One group in the north of the country that are essentially what they have been called Bermejo or Oran virus or Andes North, and then Andes South or Andes sensu stricto, that is the virus that essentially had circulated in Chubut and the south of central Patagonia. So, and why we are focused on that? Well, because in that area there had been two outbreaks, one in the city of El Bolsón and the other in the city of Epuyén, where we have seen human-to-human transmission. So the first outbreak in 1996 involved 16 cases, 9 deaths, and a case fatality rate of up to 56%. This was only 1 year after detection of Andes virus, ever the discovery of Andes virus. So there were really no tools to do a proper investigation at that time. The thing that raised alarms was that one of the cases was imported that was coming to Buenos Aires and was causing a nosocomial infection. Well, his physician, his doctor, became a case. That was the first time ever that the paradigm of the then human infections for hantaviruses was challenged, and obviously that brought a lot of attention to the issue. So since then, a lot of small outbreaks had been tried to be studied to try to demonstrate unequivocally that there was human-to-human transmission. So these were not only in Argentina, but also in different areas of the country and in Chile. So finally, in 2018, in the city of Epuyén, as I have mentioned, there was detected first an outbreak with 34 cases. Of those, they were all linked, and with 11 deaths and a case fatality rate of around 30%. And the epidemiological wave of cases demonstrated initially that obviously we have a suspicion that this was, again, person-to-person transmission. At the time, I was working at USAMRIID, and we offer our assistance to the Anlis Malbrand to support to do a full genomic investigation. And at that time, essentially, we were first able to identify that the virus that was causing the event was Andes virus in itself. We were able to sequence all of the 34 cases, get complete genomes for all of them with high quality, and with those being able to compare to first see that there was only one potential introduction, and most importantly that the variant of the virus that we were detecting was very closely related with the EpiLink 1996 strain. Is in the same cluster, in the same group of viruses. So even when they were 25 years apart, there were very few differences. Only one amino acid difference between them. And importantly, that was also intriguing that the only strain that we are being able to use in animal models for preclinical work was the Chilean strain that is highlighted in the tree. Then you can see that is an outlier in comparison with the rest. So there are 27 non-synonymous mutations between the Chilean strain that we use in our countermeasure development product and the ones that are causing that are associated with person-to-person transmission. So in the investigation of the outbreak, we essentially figured out that there were very few differences in terms of SNPs. Most of those, or almost all, with the exception of one, were synonymous SNPs. There were only one non-synonymous change in one of the branch that goes to patient 7 in the S segment. When we investigated the outbreak history along with the epidemiological information, we were able to reconstruct that there were 3 super spreaders events that actually drove the outbreak. Those were first a birthday party where the patient 1 essentially passed the virus to 5 individuals. Then one of those individuals was patient 2, who was visited by several people while sick and staying in her household, and then 6 of them were infected, and then in the wake of patient 2, one of those patients, patient 9, also transmitted the disease to another 10 patients. So by epidemiological standards, the Z value for those 3 events, 5, 6, and 10, was 2 standard deviation above the average for the rest of the cases, and that defines them as super spreaders. And the main factor, given that the virus is essentially the same, and was that behavioral ideas are actually what is driving the outbreak, the social gathering. If these patients, patient 1, patient 2, and patient 9 would have stayed at home instead of attending, events while starting to have symptoms, the outbreak would have been fully contained. So there were 4 generations of transmissions, and 4 of the cases could have different routes of transmission. So the birthday party was also an interesting event. There were more than 100 people in attendance. They were seated at different tables in essentially two different rooms. There was a very close proximity relationship between the case and the people that got infected. And one of the contacts, patient 4, only had been seated at another table at more than 3 meters, 9 feet from the index case, and had only greeted him while in the way to the bathroom. Importantly, there was a very low density of rodents in the surrounding area, and that is relevant because the index case was living in a rural area and is the one that actually was in contact with rodents, but none of the attendees to the birthday party essentially had been exposed to the same environment. So once that the outbreak were recognized, again, because this was a pneumonia case in the middle of the summer season, essentially that they did not trigger a lot of alarms with a disease that have a very long incubation period. So 6 weeks after the first case, essentially national health authorities in Argentina suggested a self-isolation in the area and avoiding social gatherings. And once that those events that was properly disseminated in the population, the outbreak essentially they are not went down below 1 and the outbreak die off. So essentially, the outbreak was caused by a single spillover event. There was no observed viral adaptation after the spillover, and that could explain the differences in transmission. And as I said, the window of infectivity was very, very short. So interestingly, this was a situation where this investigation done by a lot of people in Argentina during more than 20 years of fighting to try to get people to pay attention to the fact that this was breaking the paradigm and there was a change in the at least for Andes virus in this particular region that changed the paradigm for human to human transmission related with hantaviruses. And this obviously is a change of paradigm which always takes time. Essentially, the problem was that obviously all other hantaviruses have a dead end host historically, and they are never believed to spread in the other ways. So they are significant confounding epidemiological factors because the long incubation period made it difficult to trace what it could be human to human chains. A lot of the people evolve in the majority of the cases are living together or they are healthcare workers. So therefore it's very hard to be able to unequivocally say that they were not exposed to the same environment. The first hint, though the Buenos Aires case that I mentioned was the first claim, and the second study, the birthday party where the index case was not was in one location, and the point of exposure occur in this birthday party where the only the two events that allow us first to recognize this type of event. And obviously, without whole genome sequencing, without being able to do this fast, we would not have been able to ever recognize that these were cases of human-to-human transmission. And with that, I anticipate that my colleagues will talk about the current events. And so I thank a lot of the people that have done this, and especially a lot of the people in Argentina and Chile that had been essentially calling wolf for years regarding that this was something to pay attention to. Thank you.
Thanks very much, Gustavo, and I echo your words and the congratulations to Argentina and Chile as well for all the work they have been doing for hantavirus for years. So now it's my turn to also introduce Dr. Boris Pavlin, my colleague from WHO. He's a senior medical epidemiologist and team lead in the Unit of Epidemiology and Analytics for Response. Over to you, Boris, for your presentation.
Great, thank you. Just a minute while I put up my slides. Sorry, just a second. Having a little trouble, technical difficulty here. Okay. So I'm just going to provide an epidemiological overview of the current outbreak. So most of you will already have heard these things, that on the 2nd of May, WHO received the International Health Regulations notification from the United Kingdom regarding a cluster of severe acute respiratory illness on a cruise ship, the MV Hondius, which is a luxury expedition cruise ship that was traversing the Southern Ocean and visiting several South Atlantic islands after leaving Ushuaia, Argentina on April 1st, and was headed for Cabo Verde. There had been 3 deaths on board, and there were an additional 2 persons still ill on the vessel. And so at the time of mooring off of Cabo Verde, those 2 individuals were taken to the Netherlands as well as a high-risk contact. And at that time, a WHO and a European Centers for Disease Prevention and Control epidemiologist boarded the ship, as well as 2 additional medical officers who continued onwards with the ship to Tenerife, where on May 10th and 11th, the passengers were disembarked, and all but 27 crew members were disembarked, and after which the vessel continued on its way to its final destination of Rotterdam. During the disembarkation in Tenerife, these passengers and crew were taken on repatriation flights directly to a number of receiving countries that had arrangements to deal with their return. At the time, there were no symptomatic individuals. As of yesterday, there are a total of 11 cases, including 3 deaths. 9 of these are confirmed cases, and they are in isolation in several countries. And 2 are probable cases. 1 is in isolation in a UK overseas territory, and the other one is the index case for whom we were not able to get samples. So this is our provisional epidemic curve, which shows that the first case, the index case, developed symptoms almost immediately after the beginning of the voyage, shown on the dashed line, which means that this person was clearly exposed before joining the voyage. The following cases, the large number of cases clustering around late April there, were almost certainly exposed to the index case. And then there are 3 cases that have occurred since post-disembarkation, but of course in individuals who had exposure to the cases before the outbreak was detected and appropriate isolation was carried out. Based on our best evidence to date, you can see that we see that the index case transmitted to multiple individuals, and that there was probably another second generation of transmission after that within the ship. And there are a few still cases under investigation. New information comes every day. There was no evidence at all that any of the later cases had acquired it from rodents. And this is further corroborated by the initial sequencing information, which shows very close proximity between the different sequenced strains, again, reinforcing the notion of a single zoonotic introduction through case 1 and onward transmission through almost certain human-to-human transmission. We have also looked at where case 1 may have acquired the infection, and this is a work in progress, of course. It turns out that this individual was an avid bird watcher, and people often upload their detailed whereabouts about when they're bird watching. And we've been able to work with the company where the website that does this to understand where he went. What is still not clear, however, of course, is in this long journey, where could this exposure have happened? There are, as Dr. Palacios explained, there are many opportunities where different sub-lineages of the Andes virus are found, and it's been a bit challenging to reconcile that with the specific movements of the index case, knowing that it's the Andes sensu stricto that we're dealing with here. But I know that
We lost connection.
Hi,
Wait.
I'm back.
Okay.
Um,
Thank you, Boris.
Unfortunately, I don't think I'll be able to resurrect my slides, but I was just coming to the end anyway, which is to say that from here on out, the main issues for us as an epidemiology pillar are the follow-up of the contacts. We have on the order of 500 known contacts across multiple national jurisdictions and overseas territories, complex places to operate. Of course, it's not WHO who does the contact tracing, but we are working with member states to ensure that all the contacts, particularly the high-risk contacts, which includes everyone that disembarked the ship, are followed to the end of their quarantine period. And there are still a small number of contacts that are particularly those who disembarked prior to our awareness of this outbreak, who are still being followed, who are still being located. But as we have seen, the small number of generations that are typical in these situations, and knowing the strong measures that have been implemented to limit further transmission, we're confident that this outbreak is under control and will be brought to a close very soon. Yes, I'm not able to present my last slide, but I've finished my presentation. Thank you very much.
Thanks very much, and we appreciate very much your presentation and the time you're dedicating with all the emergencies considered. So thanks very much. And with that, I will hand over to Dr. Emma Hodcroft from the Public Health Alliance for Genomics Epidemiology, PHAGE, to take us through the panel discussion. Over to you.
Thank you so much, Josefina. And it's really my great pleasure to be here today co-hosting this webinar. I'd like to just offer my own brief introduction and kind of contextualization, because of course, the reason that we're having this webinar today is because of the role that things like genomic epidemiology can play when there are outbreaks like this one. Within days of this outbreak being identified, genome sequences were openly available, phylogenetic analyses were underway, and laboratories across South Africa, Senegal, Switzerland, and the Netherlands were coordinating in real time across multiple time zones. And of course, that kind of response is exactly what we hope for when outbreaks like this happen. And so on our panel today, we were keen to bring together the scientists that were in the room or at the sequencer, as it may be, and hear their perspective on what worked, what could be improved, and what that means for how we prepare for the next outbreak. As you already heard earlier, this is a joint webinar between IPSN and PHAGE, and I wanted to just mention what PHAGE is in case you haven't heard of it. It stands for the Public Health for Genomic Epidemiology, and it exists both for moments like this and for the times in between. It's a truly global community that's working to make genomic data more standardized, more open, and more actionable for public health. Because sequences are key, but they alone are not enough. They need to be findable, shareable, and interpretable across institutions and borders. And that's what PHAGE is trying to advance through open standards, tools, and guidance that ensure that labs and public health agencies can speak the same language, whether they're in Geneva, Rotterdam, Dakar, or Johannesburg. I'd like to just finally give a few housekeeping notes. This session, the panel section, will last for the remainder of the hour, so the next half hour. I will give some introductions, and then we will start in with the questions. For those of you listening online, you can use the question and answer function, the Q&A function, to ask questions, and you can also upvote questions. So please do this, as it's easier to monitor than just posting questions in the chat, and I will try to incorporate those into the panel. But with no further ado, I would like to introduce our panel. We have Dr. Moussa Diagne, who heads up the sequencing platform at the Institut Pasteur de Dakar, where he has built genomic surveillance capacity across West Africa and contributed directly to the sequencing response in this outbreak. We also have Dr. Jonathan Featherston, a bioinformatics scientist at NICD in Johannesburg, and the institution that received and diagnosed one of the earliest confirmed Andes virus cases in this outbreak. We have Professor Isabella Eckerle, who directs the Center for Emerging Viral Diseases, which hosts the Swiss National Reference Center for Emerging Viral Infections in Geneva, and whose lab produced and openly shared the first genome sequence from this outbreak within days of diagnosis of a passenger returning home. And finally, but certainly not last, Dr. Bas Monique, who heads the Pathogen genomics group at Erasmus MC in Rotterdam and has extensive experience in rapid genomic outbreak response across COVID-19, Mpox, and West Nile virus, and has also sequenced a case from this outbreak, making Rotterdam, where the MV Hondius docked just a few days ago on Monday, both a scientific and a very literal frontline in this response. And I want to jump straight in with talking a little bit about the sequencing and about the sample preparation and protocol. And Musa, I'd be interested in hearing from you just as a start to ask you whether you had already existing protocols for the sample preparation and the sequencing. If you didn't, how did you get these? And did you have adequate support in getting those samples ready and then accomplishing that sequencing?
Thank you very much, Emma, and thanks again to IPSN and WHO and FAGE for this webinar, and really happy to be with colleagues and friends. Yes, we can say that hantavirus genomic is really challenging diagnostic tool because many of our viruses we sequence or diagnose, such as SARS-CoV-2, dengue, or chikungunya, are the main focus. The main reason that hantavirus sequencing is not part of a routine workflow or genomic surveillance laboratory is because we don't have like a real understanding of the burden here. So yeah, it was like we had like of course in IPD a huge experience on our viral hemorrhagic fevers. And that's why we had like at least some tools like the hantaviruses from some previously validated approaches that were on our portfolio. Hopefully that's why we were able to identify quickly. So we received the samples on the 5th of May and confirmed the case less than 24 hours, like the 6th of May. We did like first the molecular sequence diagnostic and the sequencing within the 24 hours, and confirmed with a full genome maybe on the 7th of May. So that hopefully research purposes help us to get some, with some tools that allowed us to bring back those genomics. So it was like we had like tools, but mainly hantavirus tools to make it. But now we have like a complete set for hantavirus, of course, to be ready just in case.
Thank you so much for that answer, Musa. And it's very interesting, I think, and informative to hear about how experience in other viruses can lend the expertise that's needed to respond to an unexpected situation like this. Jonathan, if it's okay, I'd turn to you next and kind of continuing on that question. Is there things that make hantaviruses harder to sequence? We know that we may not all know, they are multi-segmented genomes. So they have 3 segments, the M, the S, and the L segments. Does this pose challenges, or are there other things about the virus that pose challenges when sequencing?
Well, I think in general, sequencing, when we don't have, in South Africa, for example, it's not endemic for us to have hantavirus in South Africa. So we're of course not expecting specifically to come across it. So we have to treat all our samples in this case as clinical sort of shotgun metagenomics kind of approaches. And in that respect, we did struggle initially, especially at first with coverage and getting sufficient data. It sort of relates to the previous question, but I think over the course of about 3 days, we managed to increase the yield of hantavirus by about 1,000-fold from our sequencing run. So we had protocols in place, but we had to sort of optimize them slightly. Obviously, when you're dealing with multiple segments, for a start, it means in this case tripling the work. Everything you're looking at, you have to check, double check, triple check against multiple different segments. But I'd say from our side, the greatest challenge was just generally that at least the samples that we had very low loads. So knowing exactly what kind of depths of sequencing, etc., to kind of approach was a bit difficult, and we did this a little bit. We had to take a bit of an iterative approach of including different platforms to get sufficient coverage. But that's not necessarily so different from other pathogens that are similar. A lot of work we've done previously with, say, Mpox. But in that case, that was something we were expecting, and we had primer sets, and we could amplify our whole genomes to do that rather than than just a purely shotgun approach.
Thank you. And if I can maybe just dig into that a little bit and tie it back to the first question, because I'm not actually sure exactly when you all were in touch with each other and how you started coordinating. Was having that coordination helpful for figuring out the best approaches to use, how to increase coverage, these kinds of things? Or was this a little bit before you were in communication with each other? And Jonathan, happy to go back to you, but would also be happy if another panelist wanted to jump in here.
Well, from our side, I think a lot of this changes. We kind of, in fact, I think the main lab method we use for preparing these types of samples were initially from Prof. Palacios' lab from some time back, but then the kind of additions and changes we made, things like ribosomal removal, steps like that, we added in between. So that. I think the improvements on other aspects came a lot more from when we started collaborating on the informatics side in terms of synchronizing references and that kind of thing. But perhaps better we can open that to others.
Yeah, Musa, please.
Yeah, and I fully agree with Jonathan. They did an excellent, great job to fix their issues. And yeah, when we started to have this collaboration, we're also thinking on our IPD side to also maybe support in terms of how to provide maybe enrichment-based sequencing approaches, because we have like a strong existing capacity here for RNA virus sequencing. It can allow like unbiased approaches. So one thing that came to our mind is if Jonathan and team were not able to move forward to maybe share some reagent quickly to them, but hopefully he made it happen very brilliantly. But this is just to show that it was a really example of excellent collaboration between different labs. And congratulations again, Jonathan, for this achievement.
Thank you.
Yes, and I'm definitely hoping to come back a little bit more to the collaboration in a couple of questions. I'm going to press a little bit more on the sequencing, though, for the moment. And I turn to you, Bas. We've heard a little bit about, both the historic Ebola outbreak and then, of course, the great overview from Boris about the current outbreak. But I think particularly thinking about, how sequences play a role here, could you maybe explain to us how, with genomic sequences, we can tell the difference between, for example, multiple spillover events, multiple, times this virus came on the ship, and the scenario where it's been a sustained human-to-human transmission event, and what the data points to in this particular outbreak?
Yes, first of all, thanks for having me here in this webinar and in this panel discussion. Now, I think genomics really shown us in the past, especially during the COVID-19 pandemic, that can be really quite powerful to track and trace how the virus is spreading and how the virus is evolving during time. And it addresses also one of the key questions, like in the beginning, we wanted to know, is this one zoonotic spillover or are there several zoonotic spillovers? And because of that, we, of course, need to accurately compare the sequence data, which we obtained at several different institutes, fairly with each other, because that can be quite complex to compare this data with each other, as everyone is using their own method and their own bioinformatic pipelines. And as you heard already, not all of us are hantavirus experts. So it's also quite a new genome for us. And we also are not familiar in handling these segmented genomes and how to go about perhaps difficult parts of the genomes. So that's quite essential to properly compare it with each other. And I think we did a good job by sharing all of this information amongst each other in the group. We shared our raw sequence data and the consensus sequences to really fine-tune and track and trace the mutations and see if we can come to a common agreement in about which mutations are true and which might be due to low coverage in a certain region or other technological issues. And I think by doing that, we saw that there were hardly any mutations present in the genome of the virus, which then leads towards the suggestion that there was one single zoonotic spillover followed by transmission most likely on board of this cruise ship. As we could also see in the timeline shown by Boris, it's not likely that everyone at the same time got sick, but there was really one index case who got sick well before the other patients started to show symptoms and got sick as well. So that links towards most probably having one zoonotic introduction and then followed by human-to-human transmission on the boat. Also because of the lack of the rodents present at this ship. So that's most likely that it was transmitted from human to human. And I think that's what genomics also told us. The virus is very similar to each other. We see only one virus being introduced in the ship, and then we saw spread of this virus within the ship.
Thank you so much, Bas. And I think it is a great illustration of bringing together both the information that, of course, is not sequences, the information about when people had symptoms, when people, may have had interactions, and then using genomic epidemiology as yet another strand to complement that and to see if all of the evidence is lining up in the same direction. Isabella, to kind of continue on this vein a little bit, we are, of course, it's fantastic to have these sequences from the outbreak, but we have a relatively small number. We don't necessarily particularly have a huge background of sequences from every mouse, for example, that might be infected, and we don't necessarily have every sequence from every person who's a case either, at least not yet. What do we need to be careful of when we look at a small number of sequences? And for example, what might we not be able to tell? For example, could we use the sequences to perfectly recreate, who infected who on the ship, for example? I think that's a question I've certainly had from people. Would you mind talking about that a little bit?
Yeah. So first of all, I think what a few sequences can actually tell us, and what was in our viewpoint, one of the first and most important points was that this is a known virus. So this is not something crazy we have never seen before. And the second point that was also very important for us as a diagnostic lab, do our assays actually work? And for that, having that first one sequence was already really good for us. And that's also one of the main reasons we made it public. So I think we were lucky because our quite a low CT value, so we were able to generate a full genome, and that was actually the first thing we did. We said, okay, our assay is any problem to detect this virus, and that was great to see that it works. And on your question, so I think, well, it shows that sequencing data, even for rare viruses, can be of interest very quickly. So I think it argues for broad surveillance for all sorts of pathogens, because we never know when we will actually need them as a comparison. I mean, in this case, this virus is found across quite large areas. So of course you cannot conduct surveillance for everything, but I think having an idea which viruses are circulating are really important. And for the transmission chains, well, I think somehow it was good news that the viruses are very similar, that we almost do not see any SNPs, but it also means it looks like we cannot really trace very individual transmission chains. So we cannot say case 1 really infected case 2 and then case 2 infected case 3, because they are quite similar. But on the other hand, yeah, I mean, at least we can say this seems to be one cluster that was happening on that ship. And well, hopefully we don't see more cases, so we don't need this kind of tracing. So yeah, that are a bit my thoughts on that.
No, absolutely. Certainly, we hope that we are at the end of hearing about new cases. We can cross our fingers on that. But thank you for reiterating this. I think it is always important because, of course, there is a lot we can do with sequencing. I don't think anyone on the panel would disagree with this, or we wouldn't be here. But I think it can also be really helpful to outline, that sequencing isn't the magic crystal ball. It doesn't tell us everything possible. And again, to just harken back to the last question, it's a wonderful illustration of why it's still important to incorporate more than just sequencing, to have the other more traditional epidemiological data, contact networks, and to be able to share all of that. I'd then be really interested to come back to what we talked about just a little bit earlier. And I'd maybe start with Jonathan and Musa here, though I know that you spoke to this a little bit already. I think one of the things that has been highlighted, and I am not shy to highlight it again because I think it's such a fantastic illustration of outbreak response, is the incredible collaboration that all of you as scientists and all of your countries have done in tackling and doing quick analysis of this outbreak. And I'd be curious to hear a little bit about the backstory. How did the collaboration come about? How has it worked? Are there things that you'd really like to say, this should be carried forward a bit? How can we encourage these types of collaborations to happen more in the future? Maybe Jonathan and Musa to start, but also happy to hear from Bas and Isabella as well.
Okay, great. Probably better for one of the more senior people to address this, but certainly from our side, I know the person responsible at NICD for all our special viral pathogens is Dr. Jacqueline Vea, and she personally knows Professor Palacios very well. And we share data with him. And I mean, I'm open, please, to be corrected here, but from my understanding of this, the idea was essentially multiple labs, multiple groups were sharing data with him because people trusted him. And that's a very important point, I think, here was an element of trust. And ultimately, he was seeing information from multiple groups, which initially with low coverages, we weren't quite sure. So we decided to let's pull this together and work as a team instead of perhaps ultimately coming up with confusing answers that are disparate and don't coalesce on the same answers. It would be good for I think in the end, Prof Palacios will comment on that.
Yeah, Palacios, if you'd like to jump in, I'd be happy to hear your thoughts as well.
Sure. And I have the privilege to work with a lot of the people in this group for years. Lorenzo obviously has been a great integrator of all of the activities from WHO, and essentially he facilitated a lot of the communication. I'm glad that he joined us too, and essentially I have been working with Moza for I don't know 10, 15 years, and developing a lot of or helping and working together, developing genomic capabilities in Senegal. And essentially, yes, I mean, essentially building trust, is important, and essentially sharing is caring, let's say. And then, like we say in the group, stronger together.
And those are wonderful words indeed. Musa, would you like to jump in?
Yeah, just to say that all those things is a matter of trust. This is definitely the last main point from Gustavo. And Gustavo is really instrumental to, I would say, bring people all together within this. He managed to have it shaped the collaborative spirit of this response somehow. So in a way that we are make sure that people were felt comfortable in terms of data sharing. This is not as easy as we think. We are talking about a clear discussion about uncertainties and to have a kind of lab team. It was not like institutional team, but it was a single institution altogether. I had that feeling. So I should, and we have to analyze that. Gustavo's role was really instrumental in creating this spirit of trust, openness, and collective responsibility, because we need to have really clean and clean data for public health and research, of course. And this is really important. And that's because I know Gustav for years, and we know him, and I think most of the people here know him. So that's why I really now want to make sure that people know that it's very important that in this small world of the virology and public health, trust is really important, and that makes collaboration easier. So congratulations to all the team engaged. We are always in different social network, WhatsApp, etc. We are almost in a family now. I didn't know Isabella. Now I know her. Bas is the same thing. David, etc. So that's really fantastic, and excited to have this activity done quickly and in a smooth way. Thank you.
And I absolutely think that the point on trust and connection is so strong here. I know that in the COVID pandemic, this was one of the biggest takeaways for me, was how many scientists I was able to connect with. And those connections remain. Once you have those connections built, those are scientists that you can always go to, you can always speak to, which is incredibly powerful when it comes to response. Isabella, I saw that you unmuted. Did you want to just add something?
Yeah, well, I wanted to say what I really liked is that so from our side, we're a WHO collaborating center. So we've worked with Lorenzo already on other outbreaks before, and it was very nice to be brought in contact. And I maybe have even a bit of a provocative thought. So I think it was very nice to see that if you just let the scientists do the work, they can give you really quickly information on an outbreak. And I really like this idea that WHO brings together the people who were involved, and they just work. And then we had this very intensive weekend 1.5 weeks ago, where we communicated on all channels and were working together and produced this report that was uploaded. I think it was Sunday evening. So for me, it was just this incredible power that you have when you just let the scientists do the work and they produce data, and how well it actually works. So nobody cares which country we are from, and it's really in this, how can I say, this space that has this freedom to really focus on the science and not think of anything else. And that's something I really liked a lot. And for me, I think it can be like almost like a new standard for outbreak investigation. Yeah.
That's great to hear, Isabella. And I think it is fantastic also to show, the power of science and the power of science being able to some degree get on with science. Of course, we have to always bring that back to the real world, but there is something powerful in getting the science and then figuring out how we interpret and how we act on that. The collaboration angle is so interesting, but I'm just aware that we unfortunately cannot keep everyone here for hours while I ask even more questions about collaboration. So I'm going to push on just a little bit, but it's still related. And Isabel, I'd be interested actually to hear your take on this. I think as well as the collaboration, another thing that was very remarkable about this outbreak is that data has been shared incredibly quickly and openly. And I'd be interested in your thoughts on kind of what enabled this and what this has enabled.
That was to me the question?
Yes, if you wouldn't mind starting. Gustavo, happy to come to you next.
Okay. Yeah. So we actually had this sequence on Friday, and then for us the question was a bit, what do we do with it? So we put it on virological.org, and we were in contact and said, we would also like to put it on Pathoplexus, but there was no Hunter option the moment we had it, and then we didn't want to sit on it over the weekend. But I think having a place where we can share the data immediately so everyone can use them and still kind of being recognized as provider, I think it's very important. So, I mean, we are maybe a bit more on the reference lab side, diagnostic labs, or maybe that publication angle is even maybe a little less important for us, but still, I think you really need to have a place where others can access this data. I also like a lot this way that the community can discuss and do analysis, and there are people who can do some bioinformatic analysis, maybe that we don't do. So I think having this community aspect and the different groups analyzing the data, and for me, it's really like having them out there because it's so important. It has so many practical implications to know how does that virus look like, do our PCRs work? So that's really, for me, the most important in that aspect. Can everyone just check their assays? Because this is really a rare virus that doesn't occur in many parts of the world. And many labs probably had to set up this very quickly. So that was, for me, one of the main aspects. Yeah.
Thank you. Gustavo, did you want to weigh in on this as well? You're on mute.
Yeah, I wanted to follow up on a previous question that you asked before we move on to from the webinar, because this is really interesting. You challenged us to think about what is next, what is missing that we need to do to build this even further. And I think that there are a couple of lessons from this outbreak and from hantaviruses that could be very relevant. Right now, there is a gap between the tools that we have in genomic epidemiology to present the data, share data, etc., and the annotation and the knowledge between the sequences that are available and the taxonomy, right? So for example, for in these cases, Andes is used in two senses, in sensus lato, which essentially make a lot of the different variants of the viruses that are detected in South America that are called Andes and are annotated Andes in public databases. But they are not Andes. They are a variant from Lechuanas or Oran. And therefore, is misleading if you try when you are trying to do data analysis and you are not an hantavirus expert. I mean, you are misled by the notations that are in public databases. So what we need is efforts like Pathoplexus and others that will go not only beyond collaboration and presentation of the data, but filling that gap between the ICTV that essentially deals with things to the level of a species And, for genomic epidemiology to be really relevant, we need to be able to fill those gaps. And what it is important for WHO and these type of institutions could be, for example, to support the fact that if there are hantavirus pulmonary syndromes that are detected in South America, we need to have full genome sequences of those in order to be able to later go and identify what is the case. Right now, for example, neither for leishmaniasis, neither for Oran, there is no full genome sequence available. Anyone that wants actually to look and work with partial sequences for those will be misled, and they will start talking about potential resortments, etc., because of the lack of information. So this group could be fundamental in filling those gaps that are the things that we need to have realization of the genomic potential. Over.
Yeah, thank you so much, Gustavo. Those are excellent and really well-made points, and I think are a reminder of what can we learn. Of course, even when we have successes, we have not conquered all diseases yet. So what can we take forward, and what are the kind of action points? And it's actually a perfect segue into what will unfortunately be the last question that I'll ask today. And here I'd like to go around all of the panelists. And that would be really to just give you an opportunity to tell me one thing, unfortunately, just one thing due to time, that has really struck with you from this outbreak, from the outbreak response, from your new experience with hantaviruses, or something that we haven't covered yet that you would really like to share. And I'll go first Bas, then Jonathan, then Musa, and then Isabella, just so that you know in which order I'll call on you. And Bas, starting with you.
Yeah, I think the main thing I still want to speak about is the speed on which we took to get these sequences out, because we also got complaints that it was not fast enough and we should share it more early on, also with national stakeholders and, for instance, with the ECDC. But I think after the initial release of the first sequence by the colleagues in Switzerland on Friday, if we have put out a sequence which was divergent from that, I think that would give a wrong signal. So I think it was very good that we took the time to accurately compare all of these sequence data with each other, look at the raw data, and really be sure about which data we actually put out there to prevent more confusion in the public and in the press than that was already there. So I think we really needed to take some time sometimes before we can draw robust conclusions. And of course, sequencing is now going quite fast. You can get a consensus sequence within 12 hours after you receive your sample, but you also have to be sure that you send out the right message to the public. And you should take also the time to accurately compare your data and not to put out something which you have to revise later on. So I just want to stress that point, that we just sometimes also need to take some time and a little bit of reflection before we send out a message to the world.
A very good point, the balance between speed and accuracy and making sure that we try and strike it right. Jonathan.
I think I would agree again with what Professor Palacios said about, there's been a lot of follow-up after COVID in terms of, like, for example, instrumentation. We have a lot of if I look at our lab here, for example, in South Africa, we've had a lot of investments made in technology and instruments, but there is sometimes that gap between, say, being able to sequence and knowing, having enough specific knowledge, either from a bioinformatics level or from those who know a particular lineage or collection of lineages really well. We do need to address that gap, and I think that would be probably the main thing I would say. Over.
No, but a very important point. Thank you for making it, because I do think, there's an importance between just the technical ability, which of course is key, but then also knowing what that information that comes out of the sequencer means. Musa.
Yeah, no, I think I also agree with everybody, and I also want to acknowledge every team. It was a teamwork, basically. I want to acknowledge the importance of WHO's coordination in this response too. Like, this is quite unique. It's an outbreak involving multiple countries and multiple labs, passengers, contacts spreads across different regions. And so this convening role is absolutely critical. So that's why it's really important to have like bodies like WHO and other bodies that can support that too. And that can help really to align public health priorities. And for me, my final message is that for rare or high consequences zoonotic pathogens, we need well-prepared networks before a crisis starts. And the sequencing capacity measures, it shows that with Hondius, it was critical to be ready to make it, but also coordination. And it was really fantastic to see that scientists, sequencing teams, epidemiologists could do that all together. We need to have like the possibility to have like shared protocols, quality control, and we have like a really strict sanity check during our work. The point about metadata that show that we need to be in contact with the epidemiologist team, etc. And it was mentioned the rapid public health interpretation for people. This is how we can show that genomics could be really truly actionable. And I'm really proud, and I think that we have like it's an example, a stress test, I would say, and I hope that it will be really a landmark for the upcoming public health actions in the future. And thank you again, and it was really exciting discussion today. Thank you.
No, excellent points also made by yourself, Musab, about connections and making sure that we have the coordination, because even when we have expertise, if that is in islands, it's a lot less useful than when we can all draw on it together. Finally, Isabella, to you.
Yeah, I think having access to really specialists in hantaviruses, which is just quite a virus that not everyone has knowledge in. And I think that bringing really together the experts with the lab who really then need this expert advice. I mean, understanding this complex taxonomy and this knowledge. And it's for me, it's like you cannot know everything about all viruses, and you cannot be an expert in everything. You can be a lab that generates high quality sequences, but then in the end, to really get the best out of it, you cannot do it alone. You really need the experts on this virus. You need the epi site. You need someone to coordinate. And I think for me, that was really the success story here, by bringing everyone together and having people that we can just ask about these viruses that most of us don't have a lot of experience with.
Yeah, wonderful. And to reiterate that point, I think is a fantastic, unfortunate closing to this panel, because I certainly, and I'm sure the audience would agree, could ask you guys many more questions. Just to give, I'll hand over to Lorenzo now for a closing, but I just wanted to reiterate that I do think that all of the questions and answers and the perspective provided by both the speakers and the panelists today really highlight the role that genomic epidemiology and sequencing can play in outbreaks like this, but also that it's not just sequences. The sequences were available in days, the data was shared quickly, and all of this coordination happened, but that wasn't by magic. That wasn't just by goodwill and wishes. That's because there was real efforts put in to develop that infrastructure, to make those connections, to develop those standards and protocols, and to share them. And perhaps most importantly of all, as has been touched on today, to then make sure that we have the trust and the connections to do that working together, to augment all of those skills and to bring together a network that is bigger than any of us are as individual pieces. And with that, I'll hand over to you, Lorenzo, to close out this great webinar.
Thank you, Emma. And sorry I joined late, but this is really interesting. I think I'm not gonna stress, I mean, how grateful I am for the laboratories to have trusted WHO in coordinating this effort. I'm really grateful. I think I would like maybe to reiterate the importance of the extraordinary level of scientific cooperation between Argentina and Chile and these laboratories, because of course Argentina and Chile, they have the experience, they built on decades of regional Andes hantavirus expertise and prior outbreak experience, so they have been instrumental in rapidly sharing sequencing protocol, diagnostic workflows, reagents, and allow really multiple international teams to validate their findings in real time. So I think really this is really, for me, it shows how a cross-regional event like this one really can still benefit from a common platform that is led by the experts that have the knowledge and know how to deal with these viruses. And I think this was especially important in this context because people, didn't even know what Andes was before this. So I think, and including, reference labs, I mean, they are very well prepared, but I think they have benefit a lot also from Argentina and Chile expertise to deal with this virus. So I'm really grateful for everybody and for this collaborative model that really demonstrated how sustained regional investment, including in rare diseases that not many, that don't cause many cases, etc. How that sustained investment is important, and of course also transparent data sharing that can accelerate the public health response of these to these diseases. So thank you, Emma, and thanks everybody.
Thank you so much, Lorenzo, for that fantastic closing. I'm aware we're a little over time. I apologize, but I think it was worth getting in an extra question with the panelists, since opportunities like this don't come every day. With that, I'd just like to thank our panelists, Isabella, Jonathan, Bas, and Musa, one more time for their involvement on the panel, to Gustavo and Boris for their excellent opening talks, to WHO for helping to coordinate all of this, and for ISPN and PHAGE for making this webinar possible, and of course to all of you for attending. Thank you so much and have a wonderful rest of your day.